Hi, this is Dr. Ben Morrill. Welcome to episode 7 of Reptile Genetics Weekly. Have some more cool things to talk about today, some cool updates. First of all, we are sending out more results faster, like we talked about in the last couple of episodes. In the month of May, we're going to be doing more runs because we're doing a lot more testing, more test development for new morph tests and sex determination tests. And so that's awesome for you all that have sent samples in, because that means we will be getting your results back to you quicker. So we sent out quite a bit of the results from this latest run on Friday, Saturday, Sunday, and then the last ones will likely go out tonight. So if you had samples that you sent in over I guess it'd be about 2 weeks ago, then you'll likely be seeing those results. And I'm setting up another run now. And so a week from today, maybe next Tuesday, will be another round. So it'll be a little over a week, but then we'll have more results coming out again. So, but the really fun part to all of the results this last time, as part of the developmental testing that we're doing, I had 4 different markers that I was testing for genetic stripe, to be able to see if any of them were markers that would work for us to use as a genetic stripe test. So out of those 4 markers, one of them was bad. I threw that out pretty much right away. The second one was correct, 23 out of 24. So I did 24 different known samples. I knew whether they were no genetic stripe, they were het genetic stripe, or homozygous for genetic stripe or visual. And so out of those 24, one of the markers was right for 23 out of the 24. And then the last 2 markers were right, all 24 out of 24. So it's looking really good. So you all are the first to know. We haven't really talked about this at all to anybody else. So those of you that are following these updates, you will get these little nuggets of information first. And so genetic stripe's looking really good. I have another 100 that I'll run. Yes. Yeah. So happy. It's nice when things come together. And with Clown, they came together quick. With Desert Ghost, it's been a long time. Hypo, we had, a lot. That was majority of that was done by Dr. Seidel. But anyway, things are coming together on. I guess this is the second one that we're, and even on genetic stripe, Dr. Seidel is helping us with that as well. So we're collaborating with her some. But yeah, it's looking really good. It looks like there are 2 different markers that are working 100% of the time so far. And so we'll test another 100 on this next run, and we'll see how those look. And we might weed one of those out as a bad one. Hopefully one of them stays good. It will be the correct one for us to use. And it'll be pretty quick that we'll have that listed. But if any of you that have followed all of this stuff very closely at all, all this stuff takes time. And if there is a problem, then it could take me a while to fix the problem and find another marker that does work for over 100 in a row. So we have that over 99% accuracy. But we'll always test at least 100 and get them all correct before we list anything. So we're very, very close with genetic stripe, it looks like. And next week, the next video comes out next Monday. I hope that we will have that. We might barely miss it by a day. We might not get the results till Tuesday, but I'll do my best to get them for this update next week. So you all will get to know that. And then as long as that goes well, then we'll have that listed as a test that you all can pay for and get done on your sheds. So yeah, Kayla, thanks for popping in. How are you doing today?
I'm doing great. I'm glad to hear that things are moving along with the genetic stripe stuff. That's super exciting. I know if I kept ball pythons, I would be so excited, because I know the past few months we've been saying, oh yeah, the next couple of months, hopefully we'll have some stuff coming through. So here it is. It's awesome.
Yes, it's looking really good.
Yeah.
24 out of 24, no problems at all. That's
solid.
A great start.
Yeah. All right. Yes. So want to do today's topic then?
Let's do it. That's all I've got for the update. Yeah.
All right, perfect. So we're doing some talk about paradoxing in reptiles.
All right. So first of all, the reason why we're bringing this up is we just recently had a sample come in for sex determination and had some interesting results. I went back and forth with Jeff Wilson. He's the one that sent this picture and sent the shed in. And so we'll give him a shout out. Hopefully he gets a chance to watch this.
Hi, Jeff.
Yeah, message back and forth with him quite a bit. And it's definitely interesting to think about and talk about the genetics and what could be going on and genetic testing and what may or may not work and how it could be different in different parts of the animal. So we'll talk about all that today. So this is the animal that he sent in. This was sold to him as an import that was paradoxed. And so there's not any breeding information, no breeding background, but clearly has these paradox spots. And so paradox is something that we talk about in the herpetoculture world. As a geneticist, there's two different main ways that you would end up with an animal like this, and it would be either a chimera or a mosaic. So a chimera, well, I guess both a chimera and a mosaic, what's happening is some of the cells in the body have one set of DNA, and other cells in the body have another set of DNA, and it could even be more than two. But with both chimera and mosaic, what's happening is you have at least two different populations of cells that have different DNA, different code. And so the difference between a chimera and I keep saying chimera and paradox, the difference between a chimera and a mosaic is a chimera is those two populations of cells come from two different embryos. So a chimera is where two different embryos combine and make one animal. And then a mosaic is different. So with a mosaic, it's the cells from one individual, but something that can happen early in embryonic development where there's a mess up. So if you only have, for example, and we'll show some really cool pictures later, but for example, if at the 2-cell stage, you just have these two cells, if there's a problem, a mess up with how the DNA separates and is packaged, all that kind of stuff, you can actually lose a chromosome or lose a part of a chromosome, or something can pinch off, you lose that. And so one example in humans is trisomy 21 or Down syndrome. There are people that have that in all the cells in their body. Their whole body is trisomy 21, 3 chromosome 21, or Down syndrome. But there's some individuals that only some of their cells in their body have trisomy 21. So they're actually a mosaic Down syndrome, not a full Down syndrome. So
I didn't know that was a thing.
It's not super common, but it can happen. And it's an example in humans of a mosaic. And it's something that can happen naturally. Also, when we are doing different types of assisted reproduction, then things like chimeras and mosaics, I think I haven't looked up the numbers, but I think they can happen a little more frequently, but they definitely can't happen naturally. So that's really the difference when we talk about a paradox with reptiles and herpetoculture. We're really talking about individuals that are likely either a chimera or a mosaic. Said it again. A chimera or a mosaic. So it's just a question of whether the population of cells come from two different individuals, or if it's just one individual, but somewhere during early in embryonic development, there's a mess up in one of those cells, and then every daughter cell, so every time that splits, it will continue to have that change. So like if you have a person that's part of them is trisomy 21, that means early in embryonic development, one of those cells messed up and ended up with a third chromosome 21. And then from then on, every time that cell divides, it's going to pass that different genotype, that different extra chromosome 21. And so in the example of when you just have the two cells and one messes up, then what can happen is you get these really cool splits. So you get just a perfectly half the individual is one genotype and half is another. And like I said, we'll have, we've got some
like this.
So this is a really cool example, pretty dramatic. And Kayla pointed out earlier, even the eyes, if you look at the eyes, they're in different positions, which is pretty interesting.
Yeah.
The antenna, all
the
parts
mandibles. Yeah.
a little different, and it's split right down the middle. So that's
Mhm.
pretty cool. So that's all of these that are bilateral. They are likely. Yeah. What was the bilateral? What?
Bilateral gynandromorphs.
There you go.
So sexually dimorphic animals that basically you just have half and half on one animal.
So in these ones, one side of them is male, one side is female, is what you're saying?
Yeah.
Interesting. So not only the color and pattern, yep, example with the cardinal, same thing. That's pretty cool.
Yeah.
So not only are you seeing it in the color pattern, but it's also telling you something about the sex genetics for those cells. So that's pretty crazy.
Mm-hmm. And there's actually we mentioned that in humans as well, and how there's actually been some studies on chimerism in people as well.
Yes.
Yeah. We will relate this to snakes, I promise.
Yeah. Come back to what happened with that paradox ball python shed that came in for sex determination. So just like Kayla said, in humans, there's been some studies. Obviously, there's a lot more genetic research done on humans. So this paper is on the impact of chimerism in DNA-based forensic sex determination. And so this paper, they specifically say, and I'm just going to read this quote, but still there are situations like chimerism where an individual possesses both male and female specific factors together in their body. Sex determination analysis in such cases can give erroneous results. And so now we'll come back to the ball python. So the one that Jeff sent in, the shed gave us a sex determination of being female. And in talking with him, he had read, if it is because he was thinking since it was I could say chimera, but a paradox. So if it was a paradox, then there's a chance it could be hermaphrodite. And if it was, then it likely would come out as being a male because it would contain, a lot of the time, what will happen is you'll have two X chromosomes and a Y chromosome, and you'll end up with that different phenotype. So he was thinking that if it was a hermaphrodite, then it might come out as male. But when we did the sex determination test and it was female, and he kind of sent pictures and asked questions, and I encouraged him to find someone that could either, if the animal is big enough, you can ultrasound it and see if you can see follicles developing, and that would tell you it's a female, or someone that has experience popping older ball pythons that could do the popping. And so he was able to find a friend, Will Bird, which some of you watching this may remember. Will's been keeping and breeding reptiles for lots of years. I remember talking with him on forums 15 years back. So yeah, he popped this individual, and it did have hemipenes that looked normal, very clearly a male. And so this is an example. Potentially, we'd need to do some more testing. Potentially could do some more genetic testing, maybe sperm plugs from this individual, or actual sperm, and then dry that out and test that. And there's other things we could do to try to figure out if it truly is a sexually, have those two different sexes in this one individual. So
let's do a paper on this one animal.
Actually, yeah, that would be interesting. Then I think we had another study.
Yeah.
So here they're talking a little bit about, they're specifically talking about natural human chimeras. And in this one, they say two zygotes can fuse together during an early embryonic stage to form a fusion chimera. Most chimeras remain undetected, especially if both zygotes are of the same sex. So as you can imagine, if in a person where we don't have color and pattern mutations very often, it's extremely rare, there would be a lot of chimeras. As long as both the embryos that fuse are the same sex, then you really wouldn't be able to tell that individual is a chimera. But if you go to the next slide, So here we see that even sex discordant, back one.
Oh, my bad.
Even sex discordant chimeras can have a normal male or female phenotype. Only 28 of the 50 individuals with a 46XX/46XY karyotype were either true hermaphrodites or had ambiguous genitalia. So what this one tells us, this study and this quote specifically, this tells us that even if it is an individual that has both cells of both sexes, they can be full male or full female. It really just depends on where those cells are, and whether they affect the development of the sex organs and in turn the gametes that can be produced by that individual. So we'll talk a little bit more about that. But yeah, go ahead and show that picture. This is the one we had in the thumbnail.
Yeah.
So this is a cool baby I hatched out. You can see the split face on this one. That was a really, really cool snake. That one sold really fast.
I'm sure.
Can't remember. Oh yeah, can't remember who ended up with this one, but really cool split face. I was out of town when this one hatched, and Kevin, that's my partner here with the ball python breeding that we do, Kevin sent me a picture of it in the egg with the head sticking out of the egg with that split face. And
that must have been a really cool pip to see.
Oh yeah, always fun. As long as it's a weird thing, the animal's still healthy, and this one was very healthy and did great. So that was fun.
Excellent. Yeah. Right. Let's see. We've got some questions.
Did we? Yeah. Let's go ahead with this. Yep.
Okay. Sorry. Question that came in, I think this was a phone call conversation you might have had. If you sent in a paradox animal shed for testing, will the DNA on the paradox spot be different from the rest of that individual shed?
All right. So yeah, this is one that I've answered many times over the years. And in theory, that certainly is a possibility and would make perfect sense. I haven't personally ran parts of a shed from a paradox animal and gotten different results. I haven't tried. So it's not like I've tried and been unsuccessful. I just haven't tried. So any of you that have paradox animals, like I said, I sold that one that I had. I guess I have one other one still that I could maybe try to do that with. But if you have a really good kind of like, it's easy to tell from the shed the two different looks, the two different phenotypes.
Yeah,
that would be great to be able to send that in, and we can see if we can detect a difference in the DNA.
Yeah, that would be cool.
But yeah, that's definitely possible. So not only with sex, but that could also affect morphs. So if you have some, a normal looking ball python that has patches of albino, it could be that the normal looking part is completely normal, or it could be het for albino. And then obviously the parts that look albino would be albino genetically. And so that would be cool to test that out. But if you sent it in as a pos het shed test or something like that, I don't know for sure what the test is going to say, or whether that's, going to tell you how it's going to breed, which I think is the other question that we have.
Yeah, I think so. Let's see. We have a picture of Charm.
Yeah. This is I think this is the first ball python Kevin ever bought. So this is a female ball python that has a chunk of just GHI, and then the majority of her, she's a GHI champagne. And so this is Charm. She's one of the most chill snakes that we have. She acts like a little puppy dog. Sometimes we'll just open her cage a little bit, and she'll stick her nose out, and she'll just sit there with a third of her body out and just watch us while we're in the room. And she just is really, really laid back and just likes interacting. She seems like she wants to be a human or a puppy dog out there with us doing everything we're doing.
Best puppy dog.
Yeah. But yeah, so this is the one. We do still have this girl. So if I think about it, I might try. I guess we have to get to where we have a champagne and a GHI test. But I could at least test it for sex and see if we get two different sexes. That would be interesting.
That would be really cool.
Yeah.
Okay. And actually, yeah, it does relate to the next one. So is there a way for genetic testing to determine which genes a paradox animal can pass down?
Yeah, so this one, it's pretty difficult to know. Even with a genetic test, probably the best we could do if it's a male is we could, because really all that whole animal that you're looking at, like if you go back to the picture of Charm for a second, if you look at Charm, the majority of that animal is both champagne and GHI. And so you can kind of safely assume that most likely the gonads, the part of her body that's going to produce gametes, also be both GHI and champagne. And since I've gotten multiple, Kevin and I have gotten multiple clutches from her over the years, we know she does pass both GHI and champagne. But since there's that patch that's just GHI, there is a chance that where her gametes, where her eggs are produced, could have also been just GHI. And in that case, even though visually she has champagne, it's possible that her offspring could never get champagne because the cells in her that produce her eggs only have GHI. And so that's possible. And I think most of the time you can make a pretty safe assumption by what's the majority of the animal. More than likely, that's going to be how it's going to breed. But you really aren't going to know for sure until you breed it and see what comes out. And then with a male, if you could test the actual sperm, that's a little better because then you're actually seeing what gametes that male produces. So any of you that have a paradox male and you want to know how he would pass on his genetics to his offspring, that's a possibility to get a sperm plug or, to get some sperm and let that dry and send that in as a sample. That's a possible way. With females, and even then, I can't say we would know for sure, but that would be probably the best thing we could do to try to tell. With females, you would have to get eggs. And so by the time you're getting eggs, then they're going to hatch in 2 months. And so you just find out by breeding her. These are really cool reptile examples of or other species that have the chimera or mosaic thing going on, the paradox look.
And each of these guys should have where we got the paradox pictures from. So lots of different species. I remember seeing that hognose in a group somewhere from Bosporus years ago, and it just blew my mind. I hope they still have that animal because that is cool. So cool.
Yeah, it's awesome when they get these just big old patches. This one's really sweet. Where you got patches alternating. That's really interesting.
Yeah, that would be hard to tell from looking at it just because of how big the patches are of paradoxing. Like I don't know which side is the paradox and which one is the actual the main animal, honestly. Like that's
this one could be an example of a chimera where it's two embryos coming together and the cells just kind of shuffled, and so you're getting patches all mixed throughout, so it's kind of half of one and half of another. It's possible.
And then you just have like speckles of it all throughout, like on this boa. I kind of like this one too.
Yeah, that's neat.
And then that rainbow boa, like I don't know what's going on with that, but it's pretty.
That is crazy looking.
Yeah.
Looks like it got bleach spilled on it.
Right. And then this little white-sided rat snake, black rat snake, just has one little eyebrow. So cute.
Very cool.
And one last picture of the animal in our thumbnail today.
Yes.
Little split face guy, and I love his little green, like single patch of green eyeshadow.
Only has it over one eye.
Yeah, so cool. But yeah, that's I reckon that's all we have on paradoxes today.
Yes. Thank you for watching. I hope that's helpful. I know over the years I've been asked many times about paradoxing and what's going on, and now that we're doing the genetic testing, how that could have possibly affect. And I have had multiple conversations with people about results that seemed strange, and they were from paradox animals. And you might see some. There's a really cool example that Canova has that I think they're planning to do a video on. That'll be a fun one to see. So yeah, there's lots of cool stuff as far as that go. That if you breed reptiles for very long, and I guess probably other species as well, you end up seeing these, and they're really cool. Oh, and one other question that does come up with paradoxing is, can you breed 4 paradoxes? And so it's not like a simple single gene trait like albino or piebald or something like that. But it does certainly seem to be that some morphs do have more paradoxing happen in those individuals.
Yeah, like champagne and ball pythons, I think.
Yeah, that seems to be a little more common. I remember back in the day, the snake keeper, they had some hypo combos. They seemed to have several paradox individuals in that hypo line. I don't know if it was the hypo or if it was something else in that line, but they seem to get more. But it wasn't like you can breed one and half your babies are going to be paradox, or you can breed 2 together and 1/4 are going to be, or half. It's not something. It's definitely more rare. It's more of a polygenic thing that you'd have to line breed for if you were going to try to. But I think it'd be pretty difficult. So it's kind of like a 1 in a million. When you do get one, it's not something that's easy to reproduce.
It's very lucky.
But yeah, thank you for watching. Hope it's helpful. Please do send us comments. Comment in the video below, like and share and subscribe, all those good things. We appreciate it. Notification bell.
Yes, please.
Yes, and we do. Kayla and I talk multiple times every week about the conversations and questions, and we have lots of cool stuff to come for next week too that we're already planning on. Actually, we're kind of planning 2 or 3 weeks out, which is good for us. Sometimes it's only the next week, but we've got a lot of cool ideas from people, questions and comments, and we appreciate that.
Yeah, well, thank you guys. Yeah, like, comment, subscribe, and I guess we'll see you guys next week.
Sounds good.
All right, and outro in 3, 2, 1.